Hello, here we are with news from the website prostatewarriors.com.
Glad to be here and ready to get into it.
Yeah, we have a lot to cover. For decades, treating advanced prostate cancer has basically been like a hostage situation.
Yeah, that's unfortunately a pretty accurate way to describe it.
Right, like the cancer is the hostage taker, your healthy tissue is the hostage, and our only weapon is a sledgehammer. Or, well, a bomb.
Exactly. You just blast the whole area with massive toxicity and you just cross your fingers.
Just hoping the tumor takes more structural damage than the patient does.
Right. And clinically, that sledgehammer approach is what systemic chemotherapy has historically been. I mean, the limiting factor in oncology wasn't that our drugs couldn't kill cancer cells.
Because they could.
They absolutely could. The problem was that they were just way too toxic to the human body to use at full strength.
Which brings us to the mission of our deep dive today. We're going to explore a massive, really fundamental shift happening in the research right now.
It's a very hopeful shift, honestly.
It really is. We are finally putting down that sledgehammer and moving toward highly precise, logic-driven warfare. A smart scalpel, if you will.
Yeah, that's a good analogy.
So whether you are prepping for an oncology meeting or you are just fiercely curious about how far medical technology has come, we are going to extract the most vital insights from recent data for you.
And I think the overarching theme in today's sources is that researchers are finally solving that toxicity problem we just talked about.
Right.
We are engineering ways to hit the cancer much, much harder, specifically because we are finding ways to leave the hostage completely unharmed.
I love that. So let's jump straight in the front lines, the active clinical trials happening as we speak.
Oh.
I want to start with T-cell engagers.
Oh, yes.
Because conceptually, these are fascinating. They are molecules designed to physically grab one of your immune T-cells, drag it over to a cancer cell, and force an interaction.
Which sounds great in theory.
Right. But historically, they trigger cytokine release syndrome, or CRS. Basically, a massive, dangerous immune overreaction. Why does simply connecting two cells cause such a systemic storm?
Well, because the immune system isn't a scalpel, it's an alarm system.
Okay.
When traditional T-cell engagers grab onto immune cells indiscriminately, they trigger a cascade. The immune cells panic, they think there is a massive systemic infection, and they flood the bloodstream with inflammatory proteins called cytokines.
And that causes the high fevers, dangerous drops in blood pressure, organ failure.
Exactly. To fix this, you have to control exactly when and where the drug turns on.
Which brings us to a drug called JNX-014. It just entered a phase 1 trial for metastatic castration-resistant prostate cancer.
Right, the JNX-014 trial.
And it is a T-cell engager targeting PSMA, but it uses a double mask design. Okay, let's unpack this. I look at this like a highly classified agent with a geofence trigger.
That's a great way to visualize it.
It literally has molecular masks covering both its target binding arm and its T-cell grabbing arm. It can't deploy its weapons until it proves it is standing inside the tumor.
Yeah, the delivery mechanism is really what makes it unique.
Yeah.
Those masks on JNX-014 are heavily engineered peptide sequences, and they're designed to remain firmly in place while the drug circulates through healthy tissue. The only thing that can cut those masks away are specific enzymes called proteases.
And those proteases are only found in the tumor.
Right. They are highly concentrated in the tumoral microenvironment. Tumors use proteases like biological machetes to slice through surrounding tissue so they can grow and spread.
Oh, wow. So JN-EG014 uses the tumor's own weapons against it.
Exactly. It circulates in a dormant state, totally ignoring healthy cells. But the moment it enters the protease-rich tumor neighborhood, the masks are cleaved off.
It suddenly wakes up.
Yes, it wakes up and binds to the T cells locally.
Meaning you avoid activating the immune system systemically.
Yeah.
I mean, it widens the safety window so much that they are actually exploring potential outpatient use for a T-cell engager.
Which is incredible.
Right, because that used to guarantee a stay in the intensive care unit.
Oh, absolutely.
But there's another drug taking a totally different approach to solving this exact same cytokine problem, and that's AZD8359.
Ah, yes. The CRUS-1 trial.
Yes, the CRUS-1 trial, which is currently a phase 1/2 trial, it targets a marker called STEAP2, which is highly restricted to prostate tissue.
The targeting is precise, yeah. But what's fascinating here is the architecture of AZD8359. It's built on something called the Titan platform.
Yeah, I saw that in the notes, but what exactly does a platform like Titan do?
Think of it as a specialized biological scaffold. Historically, if you tried to build a drug with 3 different arms, a trispecific molecule, it would be unstable.
It would just fall apart in the bloodstream.
Pretty much, yeah. But the Titan platform allows researchers to build a stable trispecific molecule that can perform highly complex binding.
And why does it need to be trispecific?
Because it needs that complexity, so it can be incredibly selective about which immune cells it invites to the fight.
Okay, let's unpack this. If standard engagers cause a cytokine storm by grabbing immune cells indiscriminately, how does AZD8359 choose its fighters?
Well, our immune system relies on different subsets of T cells, right?
Right.
CD4 cells are the helper cells. They are essentially the alarm sounders. When activated, they are the ones that flood the body with those inflammatory cytokines.
Okay.
CD8 cells, on the other hand, are the actual assassins. They do the localized killing.
So by specifically targeting the CD8 co-receptor, AZD8359 physically recruits the assassins, but it completely bypasses the CD4 alarm sounders.
Precisely. It fixes the root cause of the cytokine storm by leaving the alarm untouched.
Wow.
In the preclinical models, this targeted approach dropped cytokine release by 40% while still driving complete tumor regressions.
And not just anywhere. The data showed complete regressions in tough bone metastasis models in mice. I want to pause here because bone metastases are notoriously hard to treat.
Very hard, yeah.
Why is a complete regression in the bone so much more impressive than in soft tissue?
Because the bone marrow is basically a fortress. It is the birthplace of your immune cells, and naturally the body heavily regulates that environment to prevent autoimmune disasters.
That makes sense.
So when prostate cancer metastasizes to the bone, it hijacks those natural suppressive signals. It surrounds itself with a force field that tells immune cells to stand down.
And seeing a T-cell engager cut through that specific immunosuppressive environment adds immense translational weight to this phase 1/2 trial.
It really does.
But, if we know the tumor builds a suppressive force field around itself, controlling the T-cells feels like it's only half the battle.
That's a fair point.
What if the problem is the toxic neighborhood itself?
The tumor microenvironment is arguably the biggest barrier in modern oncology right now. You can engineer the perfect T-cell, but if it suffocates the moment it enters the tumor bed, the therapy fails.
Which brings us to a drug called PTT-4256. It's currently in the RASIC-1 trial, a phase 1/2 trial for advanced solid tumors, including prostate cancer.
Right.
Now, I read the mechanism for this, and I have to push back a little here.
Okay, go for it.
Wait, so this drug doesn't actually kill a single cancer cell directly? It just acts like a neighborhood zoning committee to change the pH rules. That sounds completely counterintuitive.
I know it is a radical departure from traditional oncology, but the logic is actually very sound.
Really? How so?
Well, PTT-4256 is a small molecule inhibitor of a receptor called GPR65.
Okay.
To understand why this works, we have to look at tumor metabolism. Tumors grow so fast that they burn through energy using a messy process called glycolysis.
And that creates waste.
Exactly. This process dumps massive amounts of lactic acid into the surrounding tissue.
So the tumor neighborhood becomes highly acidic.
Yes. And that acid flips a switch. It activates the GPR65 receptor on the surface of local immune cells.
The neighborhood zoning rules change.
You could say that. Normally, the immune system has cells called macrophages, which act like the body's cleanup crew.
Right.
But when GPR65 is activated by acid, it brainwashes those macrophages. It turns them into immunosuppressive bodyguards that protect the tumor and put the brakes on your killer T cells.
Wow. So PTT-4256 acts like that neighborhood zoning committee. It doesn't drop a bomb. It just blocks that GPR65 receptor so the acid can't brainwash the macrophages.
Exactly. It normalizes the microenvironment.
Yeah.
By taking the brakes off, you are stripping the tumor of its protective force field.
Allowing your natural immune system to rush in and do its job.
You got it.
If I'm listening to this, I'm probably wondering, if acid is such an obvious problem and this receptor acts as the brake, why haven't we been targeting this for years?
That's a good question.
Does this actually work in humans, or is this just a neat theory in a petri dish?
Well, drug design is incredibly difficult. Finding a small molecule that blocks GPR65 without accidentally shutting down other vital receptors takes years of engineering.
Right, the precision has to be there.
But we know the concept works in humans because of natural genetic proof. Researchers have tracked patients who carry a natural loss-of-function mutation in the GPR65 gene.
Meaning they were born with a genetic typo and their acid-sensing brakes are naturally broken.
Correct. And those individuals live significantly longer across several different tumor types, even in cancers that usually resist traditional immunotherapy.
That is incredible.
Yeah, so this drug is simply attempting to mimic chemically what genetics has already done for a lucky subset of the population.
Normalizing the environment so the immune system can work is just brilliant. But let's step away from the active trials for a moment and look at the lab bench.
Okay.
Because if we want to program the drugs themselves to be smarter, to actually make decisions before they release their payload, scientists are having to borrow concepts from entirely different fields, like computer engineering.
Yes. The convergence of synthetic biology and computing logic is yielding some of the most exciting preclinical data we have right now.
Here's where it gets really interesting. The prime example is DNA-smart drugs, which was recently detailed in Nature Biotechnology.
Oh, this is a great paper.
They've built a molecular logic gate. It requires 2 different markers to be present on a tumor cell before the DNA strands assemble and release the drug.
It's brilliant.
It is literally 2-factor authentication for cancer therapy. You need both passwords to log in and deploy the payload.
It is an elegant solution to the problem of tumor heterogeneity. Because prostate cancer is messy.
Very messy.
The cells mutate, they hide, and they shed their surface markers. If your targeted therapy only looks for one password, the cancer simply changes it and becomes resistant.
And healthy tissue might share that one password too, right?
Exactly, which leads to severe off-target toxicity.
But requiring 2 distinct markers changes the math completely.
It exponentially increases the selectivity. The system stays entirely inactive if it only finds one marker. The DNA strands are designed to only zip together and form the actual drug delivery structure when both conditions are met. You could pair PSMA with a second surface marker to create an ultra-precise gate.
Guaranteeing the payload only drops on the tumor.
Precisely.
And speaking of payloads, even a perfectly targeted therapy is useless if the inflammatory response to the payload kills the patient.
True.
Which means we have to talk about a new spin on CAR-T cells. Standard CAR-T has been an absolute miracle for blood cancers.
It really has.
But it struggles in solid tumors like prostate cancer because the immune reaction is just too violently toxic.
Right.
But researchers have now designed a PSMA-targeted CAR-T that uses an innate immune receptor called CD16A for its internal signaling.
And this is a vital mechanical shift. Classic CAR-T designs use very aggressive co-stimulatory domains.
Okay.
Once they latch onto a cancer cell, those domains act like a gas pedal glued to the floor. The T-cell attacks with such ferocity that it causes massive cytokine spikes.
But with the CD16A receptor, the approach is milder. In mice, the median survival jumped from 71 days to 97 days.
Which is a huge leap.
And 3 out of 5 mice showed complete responses. But the real kicker is that they achieved that survival benefit without driving those extreme cytokine spikes.
Yeah, and if you connect this to the bigger picture of oncology, a milder cytokine profile isn't just about patient comfort.
What else does it change?
Well, if the therapy doesn't put the patient in the ICU with an immune storm, it changes how we can use the drug.
Right, because you don't have to just give one huge dose.
Exactly. It means we can safely administer repeated doses over time or use higher dose therapies without putting the host in danger.
So we can begin treating advanced cancer more like a chronic management protocol.
Right, rather than a one-time all-or-nothing gamble.
But cancer adapts. Let's say we target PSMA perfectly. We use the logic gates, we use the milder CRTs, and we put the tumor under immense evolutionary pressure.
It will try to find a way out.
The dark reality of prostate cancer is that it is a shapeshifter. It often just sheds the PSMA entirely, reprograms itself, and becomes neuroendocrine prostate cancer, or NEPC.
Yeah, the chameleon effect.
It basically goes off the grid. Where do we go then?
It's true. When prostate cancer sheds the androgen receptor to survive our treatments, it undergoes a radical transformation. It activates a specific transcription factor called ASCL1.
And ASCL1 flips a genetic switch inside the cell.
Yes, it opens up different sections of the chromatin and switches on the expression of a completely new surface antigen called CA-CAM5.
Okay.
So it's a reciprocal relationship. As the PSMA target disappears, the CA-CAM5 target appears.
The chameleon changes its colors to hide, but now we know exactly what color it turns into.
Exactly.
And researchers have built a theranostic pair to hunt it down using an antibody called tufamitamab.
Tufamitamab, yes.
The process is fascinating. First, they attach a radioactive isotope called zirconium-89 for PT imaging, so you can clearly see the CA-CAM5 on a scan.
Right, that's the diagnostic part.
Yeah. Once you confirm the target is there, they swap the zirconium for actinium-225 to deliver the actual therapy. And the results, I mean, I had to read the data twice.
Just pretty stunning.
In NCI H660 mice xenografts, which is human tumor tissue grafted into a mouse, representing one of the most notoriously treatment-hardened PSMA-negative models we have, it produced a 100% complete response.
Yeah, total clearance.
A 100% complete response in every single treated mouse in one of the most resistant cancer models we have. Wait, really? How does actinium-225 pull that off?
It comes down to the raw physics of the payload. Actinium-225 is an alpha emitter. Standard radiation therapies often rely on beta particles.
Okay, what's the difference?
Beta particles are like a scattergun. They travel further through tissue, but they do less concentrated damage. Alpha particles are completely different.
How so?
They deliver ultra-short range, massively lethal radiation. Think of an alpha particle not as a scattergun, but as a microscopic bowling ball.
Heavy, slow, and devastating to whatever it hits.
Precisely. The energy is deposited across just a few cell diameters. When that alpha particle strikes the cancer cell's DNA, it causes irreparable double-strand breaks.
Oh, wow.
And the crucial part is that this physical mechanism bypasses biological resistance entirely.
Because it's just pure physics.
Right. It doesn't matter if the tumor has mutated its cellular pathways to ignore chemotherapy or if it's hiding behind an acidic force field. Physics always wins. If the actinium-225 gets inside the cell, the DNA is shattered.
Chasing these future cures is vital, and the Theranostics data is just mind-blowing. But I want to pivot here.
Sure.
Because we have thousands of patients who need optimized care right now, today. We have powerful tools currently in the pharmacy. The question is, how do we use them better?
It comes down to data and patient stratification, knowing exactly when to deploy the weapons we already possess.
Right, okay, let's unpack this. Let's talk about the Cabazitaxel score. This is a new integrated prognostic score designed specifically for the chemotherapy drug Cabazitaxel.
Based on the Capacity study.
Yes. And they rigorously validated it in 2 massive phase 3 trials, Prostate and CARD.
Yeah.
It uses 6 routine variables: performance status, number of metastases, PSA, LDH, alkaline phosphatase, and hemoglobin.
And notice what isn't on that list. There is no expensive genomic sequencing, no specialized receptor mapping. It is built entirely on standard blood work and routine clinical assessment.
But the way it separates patients is drastic. If patient falls into the favorable risk category based on the score, their median survival on the drug is over 21 months.
Right.
But if they fall into the poor risk category, it's only about 6 months. So what does this all mean? Why is a simple blood test score suddenly revolutionary for a chemotherapy drug we've had for years?
This raises an important question, really. It proves that the clinical benefit of a drug is not universal. It is highly dependent on the physiological state of the patient at the exact moment they receive it.
Timing matters.
Exactly. Timing matters. Historically, heavy-hitting cytotoxics like Cabazitaxel were viewed as last resort options. But this score tells us you cannot wait until a patient is physically deteriorating to use it.
Because if you wait until they slide into that poor risk category, the drug just doesn't buy them the time it could have.
Exactly. You have to administer the therapy strategically when the patient is physically robust enough to actually reap that 21-month survival benefit.
Which forces us to use supportive care to preserve a patient's functional eligibility.
Right, rather than just throwing drugs at them sequentially until they fail.
Understanding prognosis is critical, and there is a massive new observational study on another marker called B7H3.
Oh, this one is fascinating.
They looked at over 8,000 prostate tumor samples with matched DNA and RNA data. And the prognostic value of this marker actually flips depending on the stage of the disease.
It is one of the most fascinating biological twists in recent literature.
Right. Because if you have high B7H3 expression in primary or hormone-sensitive stages of prostate cancer, it is associated with worse survival.
Yeah.
But if you have high B7H3 expression in late-stage metastatic tumors, it is associated with better survival. How does a marker completely reverse its meaning as the cancer advances?
It tells us that B7H3 isn't just a static flag sitting on the cell surface. Its actual biological function changes as the disease evolves.
Okay, so what is it doing?
In a primary tumor, it might be helping the cancer grow. But once the tumor metastasizes and sets up camp in a foreign organ, high levels of B7H3 might actually make the tumor more recognizable to certain immune elements.
Oh, interesting.
Or it might indicate a slower growing phenotype in that specific environment. This is vital intelligence because B7H3 is currently a major target for new clinical trials.
We have to know exactly what this target represents at different stages of the patient's journey.
Right. Otherwise, we might treat the right target at the wrong time.
And the study didn't just look at the molecular data either. It highlighted clinical realities, noting that Asian and Pacific Islander patients showed the poorest survival in metastatic disease.
Yes, and that was completely independent of their B7H3 expression levels.
Which is a stark reminder that mapping the molecular biology of cancer down to the receptor is only part of the equation.
Definitely. We clearly have systemic or unmapped genetic variables impacting how this disease progresses in different demographic groups.
Right.
It points to an urgent clinical need for highly specific matched cohort studies so we can understand those variables. Our advanced therapies have to work for everyone, not just the historically studied populations.
It is a crucial reminder not to lose sight of the patient behind the data points. As we synthesize everything we've covered today, a very clear picture emerges.
It really does.
The future of prostate cancer treatment isn't about finding a bigger bomb. It is about outsmarting the disease. It's using geo-fenced logic gates like JNX014 to prevent cytokine storms. It's deploying neighborhood zoning committees like PTT425V6 to strip away the tumor's acidic force field.
Exactly.
And it's using the Kappa score to smartly time the heavy-hitting therapies we already have.
It is a profound shift from brute force to elegant logic. And I want to leave you with a final lingering thought to explore on your own.
Go for it.
We talked about those DNA logic gates at the lab bench, requiring 2 distinct tumor markers today to unlock their payload. The proof of concept works.
Right, the two-factor authentication.
Exactly, but computing power and molecular engineering do not stand still. What happens in a decade when we can program those DNA strands to require 5 passwords?
Oh, wow.
Or 10? At what point does the cancer simply run out of places to hide?
That is the incredibly hopeful future we're tracking right here. Until next time, to all of you listening, stay strong and fight on.